In this video, we will discuss about eukaryotic DNA replication and what are the features and what are the enzymes present in this and what is the process behind this and what are the significance of this replication. All these things we will discuss in this video. Now, what is replication? DNA replication is the process by which an organism duplicates its DNA into another copy that is passed on to daughter cells. That is, consider this is a double-stranded DNA. This is the parent DNA. This will split like this. Just imagine that. And a new complementary strand. A new complementary strand is synthesized for this and also for this strand. So we get one new strand, one old strand. Here also one new strand, one old strand. This type of replication is known as semi-conservative replication. That is, half of the DNA is conserved and this will then pass to the daughter cells. So the newly synthesized or the copy of the DNA is passed to the daughter cells. This is the replication. So the DNA replication is the process by which an organism duplicates its DNA into another copy and that is passed to the daughter cells. Now let us discuss what are the features of eukaryotic DNA replication. The features of replication. First one is replication is bidirectional and originates at multiple origins of replication known as origin in eukaryotes. That is, the site at which the replication starts is known as origin or origin of replication. Origin of replication means the site at which the replication starts is known as origin or origin of replication. In the case of eukaryotes, there are many origin. Origin of replications are many. But in the case of prokaryotes, only one origin of replication. And this replication is bidirectional. And the second one is DNA replication uses a semi-conservative method that results in a double-stranded DNA with one parental strand and one new daughter strand. That we discussed earlier. That is, this is the parent DNA and a newly synthesized, this is the newly synthesized DNA. So, this type of replication is known as semi-conservative replication. That is, for each strand of DNA, one new complementary strand is synthesized. And this type of replication is known as semi-conservative replication. That is, this contains one parent strand and one new strand. And it occurs only in the S phase. That is, the DNA replication occurs only in the S phase of the cell division. And it takes place in the cell nucleus. And the next feature is it's synthesized only in 5' to 3' direction or 5' to 3' direction. Synthesis only occurs in the 5' to 3' direction. And individual strands of DNA are manufactured in different directions, producing a leading strand and a lagging strand that we will discuss later. The lagging strands are created by the production of small DNA fragments. They are known as Okazaki fragments. They eventually join together to form the lagging strand. So the lagging strands are first created by the production of small fragments. They are known as Okazaki fragments. and they will eventually join together to form the lagging strand. And eukaryotic cell possess 5 types of polymerases which involved in the replication process. 5 types of different polymerases are involved in the eukaryotic cell replication. Now let us discuss what are the enzymes present in the replication process. First one is helicases. Helicases are the enzyme. They unwind the DNA helix at the start of the replication. Helicases which unwinds the DNA. That is, the DNA is like this. So, the helicases, they unwind the DNA like this. They break the hydrogen bonds between the strands and they make the replication bubble like this. So, they unwind the helix, double helix. And next one is SSB protein, single strand binding protein. This bind to the single strand of the unwound DNA to prevent the reformation of the DNA helix during replication. That is, they will bind to the unwinded DNA to prevent the rejoining of the helix. Next one is the most important enzyme, DNA polymerases. Eukaryotic cell contain 5 different DNA polymerases. They are alpha, beta, gamma, delta and epsilon. DNA polymerase alpha and delta replicate chromosomal DNA. DNA polymerase epsilon and beta repair the DNA, and DNA polymerase gamma replicates mitochondrial DNA. So the alpha and delta replicate chromosomal DNA, and gamma replicate mitochondrial DNA, and beta, epsilon are repair the DNA. And DNA polymerase delta synthesize the leading strand. DNA polymerase delta synthesize the leading strand. Then next one, the telomerase. That is also a DNA polymerase which contain an integral RNA that act as its own primer and which is used to replicate the DNA at the ends of the chromosome. So at the end of the DNA, chromosome ends, the DNA is replicated by the enzyme telomerase. This is also a DNA polymerase. Next one is DNA topoisomerase 1. This enzyme relaxes the DNA double helix during replication through the formation of a nick in one strand of the DNA. That is, the DNA topoisomerase 1 which relaxes the DNA by giving a nick on a single DNA strand. But DNA topoisomerase 2, which is another enzyme, it relieves the strain on the DNA double helix during the replication formed by the supercoils in the helix through the creation of the nicks in both strands of the DNA. So the DNA topoisomerase 2 relieves the supercoils formed during the replication through the creation of the nicks in both strands of the DNA. That is, DNA topoisomerase 1 forms a nick only in one strand of the DNA, but DNA topoisomerase 2 form or create the nicks in both strands of the DNA. Next one is DNA ligase, which forms a 3'5' phosphodiester bond between the adjacent fragments of DNA. DNA ligase forms 3'5' phosphodiester bond between the adjacent fragments of DNA. Now let us discuss the process of eukaryotic DNA replication. DNA replication is a fundamental genetic process and that is essential for cell growth and division. So the replication of each linear DNA molecule in a chromosome starts at many origins that we discussed earlier. That is, 1 every 30 to 300 kilo base pair of DNA depending on the species and tissue. That is, there are many origin of replication or many start points in the eukaryotic DNA for the replication, and on every 30 to 300 kilo base pair of DNA there will be one start point, and that will be depends on the species and tissue, and the process is bidirectionally from each origin. Now let us discuss the process in details. This is the double-stranded DNA. This is the double-stranded DNA. In eukaryotes, there are many origin of replications. There are many sites for the origin, for the start of the replication. So here let us consider this is the origin of replication. Here in this DNA there are 5' end, then 3' end, then 3' end and 5' end. So the origin of replication is here. Let us consider that the origin is here. Here first the helicase enzyme binds to this origin, and when the helicase binds to the origin of replication, the helicase will broken down the hydrogen bonds between the bases in these strands. The hydrogen bonds are broken down by the helicase. So the DNA become unwind at this position. So it will be like this. So here the helicase bind and the DNA, the hydrogen bonds in the DNA broken down by the helicase, and the DNA become unwind. Now the SSB protein, that is single strand binding protein, binds to each strands of the DNA. This is to prevent the rejoining of these two strands of the DNA. That is, both strands are separated by the helicase, and the rejoining of these strands are prevented by the enzyme, by the protein SSB, single strand binding protein. Now we have a DNA like this. This is the single strand binding protein SSB, and this is the helicase. So the helicase, when the helicase moves, it will broken down the hydrogen bonds between the strands, and it's unwind the DNA. At the same time, the SSB, which binds at each strand to prevent the rejoining of these strands. Now we want to add the nucleotides. For the nucleotide addition, we require a primer, that is an RNA primer, that made by the enzyme DNA polymerase alpha, which carries a primase subunit. So the RNA primer, a short fragment of RNA which is used as the primer, RNA or DNA which is used as the primer, they are made by the DNA polymerase alpha. So the DNA polymerase alpha form a primer, and that binds to the origin. This is the primer. The DNA polymerase alpha forms the RNA primer, and which then continues the addition of the nucleotides by DNA polymerase delta. So first of all, the RNA polymerase alpha synthesizes the RNA primer, and to this the nucleotides are added by, or it is continued by, the DNA polymerase delta, and the direction is always 5 prime to 3 prime. The newly synthesized strand is always in the 5 prime to 3 prime direction. So here this is the leading strand. This is the leading strand. Leading strand means which is continuous strand. That is, it require only one primer. It require only one primer. But if we look at the other strand, that is in the opposite direction. That is in the opposite direction, 5' to 3'. So the synthesis always takes place in this direction. So the synthesis starts in this direction. So the DNA polymerase alpha here also form a primer. Then the nucleotide nucleotides are added by the DNA polymerase delta. Then again, as this is discontinuous strand, the DNA polymerase alpha add the primers alternatively. So it requires many primers. This is the lagging strand. It requires many primer because the direction is opposite. So the nucleotides are added in the opposite direction. So now the final look is like this. That is, this is the double stranded DNA, and in this one is helicase, and there is a single strand binding protein which bind to each strands when the helicase unwind the DNA. And next one is DNA polymerase alpha. DNA polymerase alpha forms the primer, which is a short RNA or DNA segment, and this primer is added by the DNA polymerase alpha in both strands, but the direction is opposite. This strand is in the 5 prime, this strand is in the 5' to 3' direction, but this is in this direction. The synthesis is from in this direction. So the synthesis of this strand require many primers because it is discontinuous strand, and this one is the continuous strand and it requires only one primer. So first of all, the primer is added by the DNA polymerase, this, and polymerase alpha, and this DNA polymerase delta continues these strands by adding the nucleotides continuously. But in this case, the RNA primer is added first, then the nucleotides are added by the DNA polymerase delta, then again the RNA primer is added by the DNA polymerase alpha, then the nucleotides are added by the DNA polymerase delta. This is a discontinuous process. Here, this is the discontinuous process, but this is a continuous process. So this will continue in both strands. The DNA polymerase alpha and delta added the nucleotides. So as it is the continuous strand, this is known as leading strand. And this is the discontinuous strand, so this is known as lagging strand. So the first one is leading strand and this is lagging strand. Lagging strand is a discontinuous strand. So in the next step, all these primers are removed. So the primers are removed by the exonuclease activity. Primers are removed by the exonuclease activity of the DNA polymerase. and forming strand like this. So we get alternate strands. These fragments are known as Okazaki fragments. These short segments of DNA are known as Okazaki fragments. Okazaki fragments. And this gap is filled by the DNA polymerase delta. These gaps are filled by the DNA polymerase delta, and again here there will be some nicks, and that nicks are sealed by the enzyme DNA ligase. And now coming to the action of topoisomerase. When the DNA helicase unwinds the DNA, here, in both sides of the DNA, there will be a stress is formed due to the unwinding of this DNA in both these sides. So when helicase when it moves, the stress will increase in both sides. So to decrease the stress or to relax the stress, the topoisomerase 1, there are two types of topoisomerase 1 and 2. The topoisomerase 1 makes a nick in one strand of the DNA to relax the DNA. Similarly, the topoisomerase 2, which relaxes the supercoils formed during the movement of the helicase in the replication and by giving nicks on both strands of the DNA. So the topoisomerase 2 makes nicks on both strands of the DNA, but the topoisomerase 1 only make nicks or create nicks only on one strand of the DNA to relax the DNA. These are the main processes in the replication. I will explain once more. This is the double-stranded DNA. Five prime, 3 prime and 3 prime to 5 prime end. So first of all, consider that this is the origin of replication. Here the origin of replication also known as ori C. There are many origin of replication in the case of eukaryotes and only one origin of replication in the prokaryotes, and the replication is in bidirectional. So from each origin the replication is in bidirectional. So first of all, in the origin of replication, the enzyme helicase bind. Then when the helicase binds to the origin of replication, it will break the hydrogen bonds. In the helix, double-stranded helix, and forming a replication bubble like this. And so when the helicase unwinds the DNA, the single strand binding protein known as SSB binds to each strands of DNA, which is mainly to prevent the rejoining of these two strands. Then we require primer. Before that, when the helicase moves in this direction or in this direction, a stress is formed in both direction. Stress is formed here and here. So to relax the stress, the topoisomerase 1 and topoisomerase 2 act on these stresses. That is, the topoisomerase 1, which creates a nick on one strand of the DNA and relaxes the DNA from the stress. Similarly, the topoisomerase 2, which relaxes the supercoils by forming nicks on both strands of the DNA. Nick on both strands of the DNA. Now, let us discuss the process. So, first of all, we want the primer to add the nucleotides. The helicase unwinds the DNA like this, and we want a primer now to add the nucleotides. So, the primer is synthesized by the enzyme DNA polymerase alpha. DNA polymerase alpha. The RNA primer means it is a short segment of RNA or DNA. It is synthesized by the DNA polymerase alpha, and that have a primase function. So a short sequence of DNA or RNA which act as a primer is added at the origin of replication by the enzyme DNA polymerase alpha. Then the replication is always in the 5' to 3' direction. Always in the 5' to 3' direction. So both strands are in opposite direction. So the movement of this strand, leading strand, is continuous. That is, the DNA polymerase delta now add nucleotides to these primers continuously. At the same time, in this strand is discontinuous strand. So the nucleotides are added in this direction. So it requires many primers. That is, one primer is added by the DNA polymerase alpha, then DNA polymerase delta added the nucleotides, then again another primer is added by the DNA polymerase alpha, again the DNA polymerase delta added the nucleotides. In this way, alternate primer and nucleotide sequences are formed. And these short sequences of DNA, not the primer, and note that sequences of the DNA are known as Okazaki fragments. Okazaki fragments. And all these primers are removed by the exonuclease activity. Exonuclease activity. All the primers are removed by the exonuclease activity. And that gap is again filled by the enzyme DNA polymerase delta. Delta again fill that gap by the enzyme DNA polymerase delta. And again the nicks formed here are sealed by the enzyme DNA ligase. So we get two copies of DNA from one single stranded DNA. So we get from one single stranded DNA we get two single stranded DNA. This is the process of replication. Now let us check the points. The leading strand is synthesized by the DNA polymerase delta, and the leading strand is synthesized continuously in the 5' to 3' direction, while the lagging strand is synthesized discontinuously in the 5' to 3' direction through the formation of Okazaki fragments that we all discussed in this. At the completion of the synthesis, DNA ligase seals the breaks between the Okazaki fragments. as well as around the primers to form continuous strands. Now let us discuss about DNA proofreading. What is the DNA proofreading? In eukaryotes, only the polymerases that deal with the elongation, that is delta and epsilon, have proofreading ability. That is 3-5' exonuclease activity. If an error is detected, the erroneous base is removed via 3-5' exonuclease activity and replaced with the correct base. That is known as excision repair. Base excision repair. So, the DNA polymerase delta and epsilon have the proofreading ability in 3-5' exonuclease activity. So, if error is detected by these enzymes, they will remove that base by the 3-5' exonuclease activity and replace with the correct base. And that process is known as excision repair. They also remove pyrimidine dimers formed by the UV rays or other mutated bases and replaces them. Now let us discuss the significance of eukaryotic DNA replication. DNA replication is a fundamental genetic process that is essential for cell growth and division. And the DNA replication involves the generation of new molecules of nucleic acids, DNA, crucial for life. And DNA replication is important for properly regulating the growth and division of the cells. These are all about DNA replication. Thank you.